the last few decades, there's been a real understanding that cancer is an evolutionary process, that cancer is an evolutionary disease. Cancer is what happens when multicellularity breaks down, when conflicts arise between cell lines within an individual organism, when natural selection starts to act on proliferating cells. And cell lines can proliferate to the detriment of the organism containing them. Cancer is, of course, a hugely important medical problem in humans and other organisms, and particularly in an aging population, as cells have more and more chance to turn cancerous. So cancer is a levels of selection problem. There's selection at the level of the cell, where the cancers proliferate more and more, and there is selection at the level of the individual for anti-cancer adaptations. So many species have immune defenses against cancers, or for example, the fact that our somatic cells do not have telomerase and are unable to proliferate infinitely is thought to be an anti-cancer adaptation. So instead of horrible pictures of gory cancers, I've chosen some cute animals here because I wanted to make the point that in general, cancers are doomed. If a cancer cell line is successful enough, it will kill its host and then it too will die, with, as of about 2 weeks ago, only 2 known exceptions of cancers that outlived their original hosts, canine venereal transmissible tumour and Tasmanian devil facial tumour disease being cell lines that are able to transfer from individual to individual. So there are these 2 examples of highly degenerate mammals that are now parasitic on other members of the species. And last week or so, a clam was also found to have a selfish cell lineage that was transmissible between individuals. So cancer is a sort of an example of the short-termism of evolution. The cells proliferate now because they are unable to know that they are dependent on the host organism for survival. Another burgeoning field over the last 20 years or so has been experimental evolution. Evolutionary biology was, of course, very largely an observational science until relatively recently. But the very famous Lenski experiment, which these 12 flasks represent, and many others like it, show that evolutionary biology can now be very much an empirical science. You can take organisms, nearly always microorganisms, you can impose selection on them in real time and you can watch the results, you can watch evolution happening. This is great, particularly because you can have controlled, replicated experiments allowing you very easily to infer causality in evolutionary processes. So we thought we would try to bring these 2 strands together and start to use experimental evolution to understand cancer evolution. So at first, we were all very, very confident about how easy it was going to be to transfer the techniques of experimental evolution to cancer evolution, because you can grow cancer cells in culture in the same way that you can grow bacteria in medium. You can have cancer cell lines, you can order them from repositories, any kind of cancer you'd like, and you can, like with bacteria, freeze down your cells at any point so that you have the ancestor to refer back to. If you have your evolved and ancestral strains, you can compare them relatively directly. So those were the similarities between the 2. And of course, we quickly found that some of the differences were very important as well. Cancer cells are hugely fussy. They grow very slowly compared to microbes. We're talking about 1 day per cell generation of cancer cells. You need to grow them in serum. There is no way to actually make a synthetic medium so far that cancer cells can grow in. They need blood. And of course, these are very, very complex cells derived from humans. They have a human genome of gigabases rather than the megabases of the microbes. And they don't grow in a nice convenient suspension. They will grow across a surface. They're kind of clingy, needy cells. They love company. They'll stick together. They like to grow as a monolayer on the surface of a plastic bottle. But if you give them a non-stick bottle, they will hug together into clumps of about 1,000 cells called spheroids. So they'll grow as spheroids, or they'll grow across a surface. And a particular annoyance is that as yet, we have not been able to find a simple competition assay. A real mainstay of experimental evolution is you genetically tag your ancestral or your evolved strains, mix them together, see who wins, and for that you need essentially a selectively neutral tag that you can apply to one of your lines. We were cheered by the fact that cancer biologists insisted to us that there were these totally non-toxic neutral tags that you could apply to cells, where you could just paint our ancestral or evolved cells green and mix the two together. But in some of our first preliminary data, we found this is not true. Our untagged cells are doubling every day as they're supposed to, but our green tagged cells, while they're not dying off, are not really reproducing at all. Our red tagged cells have their growth inhibited by quite a great deal as well. So we don't have a simple competition assay, and we don't as yet have a neutral way to tag these cells. We're now experimenting with something with the amazing name of nanodots, which are inert particles that the cells apparently will take up and distribute to their offspring without doing anything to them. So we'll see whether those two are only non-toxic rather than neutral. So we wanted to choose a step in the evolution of cancer that we could examine in this experimental kind of a way. And this is a list of the important, essentially, steps in the evolution of a cancer that was put together by Hanahan and Weinberg in 2000 in what has, I think, become the most cited paper ever published by Cell. So what makes a normal somatic cell into a cancer is a series of steps allowing that cell to replicate in a limitless way, because it can no longer needs to depend on growth signals from the host. It is insensitive to signals from the host to stop growing or to die. It's able to call on the host to build blood vessels to bring it further nutrients. And then finally, the ability to metastasize and invade tissues. And we decided to work on a sort of trait that is a subset of metastasis, which is motility. And there are three reasons why we did that. Firstly, it's easy to measure. Motility can be just, you can watch cells move, you can measure how fast they're moving, you can see how far they spread out. And secondly, we thought there were some very interesting sort of bits of ecological theory that we could bring to bear on this. Tiffany had done her PhD on the evolution of dispersal and motility in bacteria, and so we wanted to look at the same kinds of questions using these cancer cells. Several people have pointed out that there is an interesting parallel between the metastasis in a cancer and dispersal in whole organisms. A tumour is actually very, very active in creating metastatic cells. It may shed millions of cells per day into the bloodstream. Only a vanishingly small proportion of the cells kind of shed into the bloodstream by a tumour will actually find a new place that they can lodge and grow into a secondary tumour. In the same way that of all the millions of acorns that are produced by an oak tree over its lifetime, only a very, very few are going to be able to grow up into mature trees themselves. So it has been proposed that the kinds of factors that favour dispersal in whole organisms will also be the kinds of factors that favour metastasis in a cancer. And one that we sort of honed in on was the idea of competition for resources. The theory around this is vast, but a lot of the thinking boils down to, if you are competing for resources in a place, then it's probably better to go away and try and find somewhere else, particularly if you're competing for resources with your close kin. So an acorn that leaves this oak is leaving resources behind for its relatives. It maybe has a very small chance of finding a good place to grow elsewhere, but that's better than nothing. So we wondered whether we could start to control or select for motility in these cancer cells by manipulating resource availability and trying to introduce competition for resources. So this was sort of the basis of our experimental setup. We started with some cancer cells called MCF7. These are breast cancer cells. They do move, but very, very slowly. We grow them. This is Anna, our PhD student. She grows them in a monolayer on the bottom of these culture bottles, transferring them every week into fresh medium as the old medium gets exhausted. And then eventually she will grow them into a spheroid in a non-stick bottle, and then allow that spheroid to splat and glom onto a surface and see how fast the cells will spread out over the surface as a measure of their motility. So we introduce low nutrients into this medium, so 0.5% serum. It's a very sparse diet for these cells, kind of Weight Watchers type stuff. And we see if this causes cell-cell competition and selects for motility. We are fairly hopeful that this medium is not entirely homogeneous and that small patches of depleted nutrients can occur. We've done some tests where we sort of spot some dye onto the medium and we see that it doesn't spread out completely all over everything. There is variation in the concentration of the dyes at the right kind of scale that the cells could move into areas of higher or lower nutrient. So we start our cells out in two lines, in a high and in a low nutrient environment, and we hope that we will see changes to their motility. And the first problem is that, yes, we see immediate changes in their motility, far too soon to be the result of selection. If we put our spheroid, so these are spheroids here, onto the surface, if they've been grown in low nutrients, they spread out very, very fast, whereas if they've been grown in high nutrients, they spread out relatively slowly. So there's a very strong plastic response that's not caused by selection. In the longer term, we see what we would have predicted, which we're quite pleased with. In the low nutrient lines, the motility, as measured by how fast that spheroid spreads itself out over the surface, shoots up. So that's 12 weeks, each week being 7 generations. And the high nutrient line, this drops down as well. So this is based on 3 replicates of each. So we do see a long-term response to the changes in nutrients. However, I don't think we have as yet nailed down that this is a response to selection. It is a heritable change. We've done essentially a common garden experiment here, where we've taken the cells that were evolved in the high and the low nutrient environments, and we've grown them in the environment they're used to, or we've swapped them into the new environment. The cells that have been evolved in the high nutrient stay at basically the same motility level when you put them in the low nutrient environment. But the cells that have been evolved in the low nutrient environment have become kind of sensitive to nutritional conditions, and they run about when they're put in low nutrients, and they sit tight and enjoy the good times if they're put in the high nutrient conditions. So we do see a sort of sustained gradual change in motility dependent on whether they're put in the high or the low nutrient environments. But we still can't be totally sure whether this is actually the response to selection. Remember, these are human cells. They have cell differentiation pathways. They have very, very strong capacity for epigenetic change, for permanent switches in gene expression in a way that bacteria simply don't. And so we'll have to be very, very careful to rule out that this is just an epigenetic switch. Or indeed, we should consider that it might be in some ways more interesting that what we're seeing is a pre-programmed response to low nutrients rather than a selective response. So we've been trying to go very much back to basics and look at, do we have everything here that natural selection needs to function? Obviously, natural selection needs heritable variation in the trait, and then we needed to see differential survival or reproduction dependent on the trait, and the trait being, in this case, motility. So this is how Anna has been measuring the motility of individual cells over time. This will repeat itself in a second, but the red line here is a mother cell. She's going to stop about here and split into two cells, the blue and the green, which themselves chase them, sort of run about on the surface here. So we can measure the speed, and also we tried measuring the sort of directionality of these cells, but we stuck with speed in the end at this sort of individual level. And we can see, basically, is there a strong correlation between the speed of the mother and the daughters, or between the two daughters, to get a kind of idea of a kind of upper bound on the kind of heritability of cell speed. Whether you look at mother versus daughter cell, or whether you look at the two daughter cells resultant from a division, there is an extremely high correlation between the speed of the one and the speed of the other. These slopes are about sort of 0.85 down to 0.65. But we have not taken these cells out, randomized them, and put them back in exactly the right environment. So while, again, this is consistent with there being a kind of genetic or epigenetic or otherwise heritable component to speed and motility, we don't know how much of this is due to variation in nutrient levels across different areas of the petri dish, or indeed just the nutritional state of the mother cell. What we are trying to do at the moment, and what I hoped we would have enough data for me to present today, is following these cells over more than one generation, setting something up over a sort of 2 to 3 days, following a cell, its daughters, its granddaughters, its great-granddaughters, and seeing whether, if you compare a granddaughter with a mother cell or a great-granddaughter with a mother, you get a shallower slope here, and getting maybe closer to the actual level of heritability of motility here. Being able to follow those cells over several generations would also allow us, hopefully, to detect differential survival and reproduction. We can see if it's the fast cells that manage to reproduce more, or if the slower cells just tend to stop and die off. And we're sort of hoping that we'll be able to look at that in the same kind of a way as you would look at individuals in a long-term field study, follow them over the generations, look at their lifetime reproductive success, and so on. So I'd be very, very interested to chat to anyone who's got sort of ideas about where we should go with this. But so far, we do see what appear to be stably heritable changes in cell behavior that are consistent with the effects of natural selection due to competition for resources, favouring more motile cells. But we don't know if it could just be a heritable plastic response. And I do think that question is interesting whichever way it is answered. And what is the importance of selection on a new mutation that causes motility in a cancer cell versus a kind of pre-programmed response to low nutrients that would just cause cells to become more motile through some kind of epigenetic or metabolic pathway. If it is the case that this incredibly important transition from a sessile to a motile state that causes metastasis, and therefore that's the step in cancer that kills you. If this is a pre-programmed response rather than a selected mutation, then perhaps it would mean very different things for the treatment of cancers and the prevention of metastasis. If it's a very careful response to environmental conditions, you might be able to change the environmental conditions inside a tumour to disfavour motility and metastasis. So, as I say, I'd be very, very keen to discuss this with anyone. And I'll finish with acknowledging the team. I mentioned Tiff Taylor and Anna. Phil Dash is a cancer biologist, and two sort of microbiologists, experimental evolutionary biologists who've been very, very important in helping bounce ideas around, have been Rob Jackson and Mike Brockhurst. I'd also like to thank the Leverhulme Trust, who funded an entirely different project, but we've managed to spend some of the time on this as well. And also the University of Reading's Europe scheme, which allowed Anna to do a summer on this before her PhD and give us some extra preliminary stuff. So thank you very much, and happy to take any questions.
Okay, thanks a lot. Fascinating. Not somebody who normally thinks about cancer and these sort of things. And I saw somebody was tweeting and saying, "Oh, this looks really good." So, are there any questions?
Thank you. Yes, very interesting indeed. To what extent are the environmental conditions that you put in your cells under comparable or related to the conditions that the cells are used to being under?
In this particular experiment, it's the easiest one for us to do. We do think, and there are differences in nutrients across spatially in a tumour. It's not often thought that sort of serum is the most important one, but growth factors might be, and those are in, no one really knows what's in serum, but it's thought that there are sort of scarce proteins in there that are very, very important. We're also measuring the same kind of thing, but tweaking acidity and oxygen levels. Oxygen is one that seems to be particularly important. Oxygen can only diffuse about 1 millimetre into any tissue. And so when a tumour gets the size of about 1 millimetre across, the cells in the middle either have to die or move or start to call for blood vessels. So basically for experimental ease, we chose this one. It's probably important, but it may not be the most important. It'd be interesting to see what the anoxia shows instead.
Do you have any more questions? Okay, well, that's good. I've got one, which I was actually curious about. Mm-hmm. So you've got your, you can look at the mother cell, the daughter cells, mm-hmm. and the problem is the daughter cells could be in the same environment.
Yeah.
But can you pick out the daughter cells, put them in different environments, and then grow their offspring again in different environments? So just try and replicate the treatments. I
think it is possible. Yeah. The problem, yeah, the problem would just be getting the right equipment and getting it to work. It's probably doable, yes.
Yeah, just needs that little lab-based skills.
Yes, yeah. Mm-hmm. Another thing we're hoping to be able to do is basically select directly rather than indirectly for motility by dropping the cells onto a bit of gel in the medium, and then later pulling off the bit of gel and throwing it away. So we only get the ones that have, explored and run away into the playground.